Hello, welcome to the Medical Licentiate Emerson channel. So today we are going to start our General Embryology Lesson 1 in Human Anatomy and Physiology combined. So, but specifically we are going to go through gametogenesis, ovulation to implantation. So this is more of after gametogenesis, then we are going to do the week 1. Okay. So in our introduction, we are going to learn that the stages of development are actually divided into 3 parts. You have the pre-embryonic period, which is 1 to 3 weeks. Then you have the embryonic period, which is 4 to 8 weeks. Then we have the fetal period, which is 9 weeks till birth. So these are the stages of development for gender embryology. Then, so how do we define embryology? So embryology is the study of the formation and development of the embryo or the fetus from the moment of its inception up to the time it is born as an infant. Then embryology is a study of the development of an individual before birth. That's also another definition. So you have two definitions. You can either define it as a study of the formation and development of the embryo or fetus from the moment of its inception up to the time it is born as an infant. Or you can say it's a study of the development of an individual before birth. Okay, so let's go back to the stages of development. Now we define them. Okay, so remember we had 3 stages of development. So number 1 is actually known as the pre-embryonic stage. Is also known as the germinal period. So this one starts from the first week to the 3rd week after fertilization, during which the zygote develops and forms the trilaminar. This will be the trilaminar, okay, and forms the trilaminar germ disc. Then in the embryonic period, so this is the first one was the pre-embryonic, also known as the germinal period. After the pre-embryonic or the germinal period, you find the embryonic period. So the embryonic period starts from the 4th week to the 8th week, during which there is a differentiation and formation of the most of the tissues and the organs of the body. So this period from 4th to 8th week is known as the embryonic period. Then we have the what we call the fetal period. So this is the last one. So the fetal period starts from the 9th week up to the termination of pregnancy, during which there is a rapid growth of the fetus and complete development of the placenta. Okay, then there are some descriptive terms. There are some descriptive terms that you need to know in embryology. So, number 1 such descriptive term is that usually, the one which we usually use, for instance, we have anterior, but for an embryo, that is referred to as the ventral, which of course is the same thing as anterior. But these are the more descriptive terms for an embryo as compared to anterior. Then for posterior, the more descriptive term is the dorsal, with the same meaning, but they are the correct terminologies to use. Then we have the superior part. The superior part is referred to as the cranial part. Then the inferior part is referred to as the caudal part. So these are the right descriptive terminologies that you have to use. Okay, so in short, we are saying, remember when we're talking about animals, find that the anterior portion of the animal we usually refer to it as the ventral, because that is the correct terminology for the anterior portion of the animal side. So that's the same thing about the embryo. So we use the words such as ventral, dorsal, cranial, caudal. So these are the correct descriptive terms. Okay, then for the sections for the planes, anatomical planes, you have the median plane, which is here, then the sagittal plane, the median section, which is here, then the sagittal plane is actually on this part. Okay, then you have the transverse section, which is done here. Then you have the frontal or the coronal section, which is and I'm sure you have realized to say it's around different from when a fully developed man is standing in an anatomical position. You have the frontal, transverse, the median, and the sagittal. So I find that they are all looking different now. Okay, so that's even the way it is actually. And then these are other descriptive terms that we also need to find in there in our study during embryology. These are some of the descriptive terms. Okay, but of course we find that most of the descriptive terms are usually very common. So these ones, fine, we are going to go through them when we actually learn in details. So let's start with the let's continue with the definitions. Remember, we last ended on the definitions for the gestation periods, the gestation periods. So now we're going to define gametogenesis and ovulation. So gametogenesis is the process involved in the maturation of the two highly specialized cells of the spermatozoon in the male and ovum in the female before they unite to form a zygote is the process which we call gametogenesis. So before the spermatozoon and the ovum combine, that is gametogenesis. So gametogenesis includes both the male and female gametes when they mature. That is gametogenesis. Then I'm sure you have realized to say there is this word spermatozoon. So spermatozoon is actually singular. So meaning plural is spermatozoa, then singular is spermatozoon. That's one thing you need to realize from there. So there are two types of gametogenesis. We have oogenesis and spermatogenesis. So oogenesis is the process involved in the development of a mature ovum. Then spermatogenesis, so meaning oogenesis is for the ovum. Then spermatogenesis, of course, you are talking about the sperm cell, the spermatozoa in plural. So spermatogenesis is the process involved in the development of spermatids from the primordial male germ cells and their differentiation into spermatozoa. Okay, so that is spermatogenesis. Okay, or others will find that you can have other definitions, but this is the best one, where we say where the development of the round spermatids from the primordial male germ cells and their differentiation into spermatozoa. Okay, that is the spermatogenesis. Then lastly, we have ovulation. Ovulation is a process whereby secondary oocyte is released from the ovary following rupture of a mature Graafian follicle and becomes available for conception. That is ovulation. So when there is a stage of luteinizing hormone, there's going to be rupture of a mature Graafian follicle, which becomes available for conception. That is what we refer to as the ovulation. Then, for gametogenesis to occur, you actually need to understand chromosomes and the meiosis. For instance, we have what we call the diploid and haploid state. So, when we say diploid, it means that the number of chromosomes in the number of chromosomes are actually double, or specifically we are talking about the 46. So 46 is what we call diploid. So if a cell has diploid number, we mean we have 46 chromosomes. That's what we mean. So we are saying 46 chromosomes is the diploid number. Then half of the 46 chromosomes is what we refer to as the haploid. Okay, so hope you have understood that part. Okay, so let me just repeat. We are saying in terms of chromosomes, if a cell has 46 chromosomes, then that is a diploid cell, a daughter cell specifically. But remember, for gametogenesis usually has haploid states. So meaning before they actually join together, they are actually half that number. That's what we call haploid states. For instance, the ovum and the sperm cell each contain 23 inside the cell. So meaning we can say they are in haploid states. That's the difference between diploid and the haploid. Then you also need to know the difference between meiosis and the mitosis. So meiosis is the cell division that happens in gametes, and it is also known as cell reduction. That is meiosis. So meaning in meiosis, the number of cells, the number of chromosomes specifically, are reduced from the diploid state into the haploid state. Whilst in mitosis, it usually just maintains. So meaning meiosis maintains the number of chromosomes. So if it's diploid, it would be diploid. Okay, so that's the one thing you need to understand. Okay, so we are saying meiosis, cell reduction, mitosis is going to actually maintain. Okay, then, but of course, these are not the only differences. The other difference is that meiosis actually occurs in gametes. So meaning the sex cells, that's where meiosis occurs in. But for mitosis, it occurs in somatic cells. Okay, that's the big difference as well. Okay, that's one of the big differences. So let's move on to another difference. Remember, we said meiosis is reduces the diploid to haploid. So meaning in meiosis, the number of chromosomes there are 23, whilst in mitosis, the number of chromosomes there are still 46, the diploid in short. That's also another difference. Then let's go to another difference. So in meiosis, there is actually a production of 4 non-identical daughter cells. So in meiosis, we are saying there is a production of 4 non-identical daughter cells. But in mitosis, there is production of only 2 identical daughter cells. 2 identical daughter cells. And they each contain the diploid number. Okay. So, hope you have gotten something from here. Okay. So, there is actually wisdom behind meiosis. So the wisdom behind meiosis is that, remember, during fusion of the male and the female sex gametes, they both have to be in haploid numbers before, so that when they fuse, they are going to make one diploid state. Because if they come together, one is in diploid, the other one is haploid, then you can have different chromosomes joining together. For instance, we can have a 46 from the man, then a 23 from the mother, then that will be a 69. Now the problem is with the 69 is that you can end up having different types of pregnancies, or in other cases we find that there is an empty ovum. There's an empty ovum, then the male sex chromosomes, either the one with 23 fertilizes that one with 0 chromosomes. Then that results in what we call a partial, or specifically just going to call it a molar pregnancy. So a molar pregnancy is a type of abnormal pregnancy that results either when then incomplete, or whether when an empty ovum is fertilized by one sperm, or it can also occur such that an a viable ovum is fertilized by one sperm and it then duplicates. So meaning it goes through the process of duplication, then they become two. So someone will still end up with 69. Or in the first one where we said in molar pregnancy there is an empty ovum which is fertilized by one sperm and it duplicates. The other one to occur is if imagine the egg is empty, it does not have the 23, then it is fertilized by two sperms. So meaning there's two sperms you have that's where the 46 are going to come from. But of course that won't be a complete, or that won't be a complete molar pregnancy. It will be an incomplete, because actually even the fetus won't be formed from there. Okay, we just have grape-like vesicles. Okay, so that's the wisdom behind meiosis. Okay, but of course there are a lot of wisdoms, but this is the most common and this is the most important one. So then lastly, what is the risk on meiosis with age? Risk of meiosis with age. So we are going to start with the female reproductive age related risks. So number one, for females, we find that they can have chromosomal abnormalities. So what do we mean by that? So we mean specifically their offsprings. So this is age-related risks. Okay, so number one, chromosomal abnormalities in females. So meaning their children might have like Down syndrome, Turner syndrome, Klinefelter syndrome. Okay, that's the first difference. That's the first risk. Then the other risk is there can be an aneuploid. So there's an abnormal chromosome number. The third risk is miscarriage. So remember, sometimes, most of the times, the body can know the structural and the chromosomal makeup of the fetus. So if it's find that it is an abnormal pregnancy, the body can just cause an abortion. Okay, so that's the miscarriage we're talking about. Okay, so that's one of the risks on meiosis with age. Then the fourth risk is failed fertilization or implantation. That's also one risk. Remember, like we said, if there is a chromosomal or structural abnormality, and of course, it might occur that there's going to be failed fertilization or implantation. Then the other is birth defects. So that's the last risk on females for their offspring, to be specific. So for instance, the children might have heart defects, cleft palates. Okay. Then for males, let's go to the risk on meiosis with age for males. So male reproductive age-related risks. Number one, decreased fertility. Find that yes, men don't necessarily stop bearing children, stop fathering children, but there is actually a decreased fertility within age. Then number two is there's going to be an increased risk of genetic mutations. Increased risk of genetic mutations. Then there's also going to be birth defects as well. For instance, autism and schizophrenia as well. Okay. Then the fourth one is that also aneuploid. Aneuploid is when there is an abnormal chromosome number. Then five, you can have increased risk of miscarriage as well. So meaning the offspring might also have an increased risk of miscarriage. Okay, so the age which we usually talk about, we find that for women is 35 and above, then for men is 40 and above. But increased risk for birth defects and aneuploid is 45 and above. Okay, so that's what you need to know. So those are some of the things you need to understand about the risk on meiosis, or the risk that comes with meiosis as you age. Okay, so we are done on the risk on meiosis with age. Okay, so next we are going to look at the male and the female gametes. So this is a sperm cell, this is an egg cell, the ovum. So we also need to know about the ovum is that, and the egg cell is known as the oocyte. Hence its production is known as oogenesis. Then there's what we call a follicle. So what's the difference between a follicle and an oocyte? So those are some of the things you need to understand. So the follicle and oocyte are two closely related but distinct structures in the female reproductive system. So number one, let's start with the follicle. A follicle, number one, is a fluid-filled sac within the ovary. Number two, the follicle is one which contains and nurtures the developing oocyte. This is the most important difference. So what we are saying is just that if this is an oocyte, then the oocyte is of course inside, but the follicle is one which is encompassing the oocyte. So meaning the outer uh, sac. This sac is what we call the follicle. Then inside the follicle, that's where you find the oocyte. That's what we are telling. That's what we are saying. So the follicle is a fluid-filled sac within the ovaries that contains and nurtures the developing oocyte. The other difference is that the follicle produces hormones such as estrogen and progesterone, which of course also even regulates the other function of the follicle is of course after these hormones are produced, it even regulates menstrual cycle. Then the last function is that it also its growth and development is stimulated by the follicle stimulating hormone. So that's the other difference. Okay. Then let's go to the oocyte. So oocyte, let's go to the oocyte. So oocyte does not produce hormones. So meaning we say the oocyte, the follicles produce hormones, but here the other difference is that the oocytes don't produce hormones. So let me just repeat the key differences. We are saying number one key difference is in terms of function. The follicles support oocyte development, whilst the oocyte carries genetic material. Okay, then the second function is structure related. So the follicles is just a sac, whilst the oocyte is a cell within the follicle actually. Okay, then the location. So the second is like structure. The follicle is a sac, oocyte is a cell. The third is location. The follicle is within the ovary. Then the oocyte is within the follicle. Then last key difference is the hormone production. The follicles produces hormones, whilst the oocytes do not produce hormones. That's also another difference. Okay, so we are done with the differences. Let's start with the spermatogenesis. So remember our reproductive lecture, we said spermatogenesis occurs in the seminiferous tubules. Okay, specifically that's where the sperms are produced. So spermatogenesis begins at puberty. What happens at puberty for spermatogenesis to begin? So that's when the solid seminiferous cords acquire a lumen, becoming tubes. So, I mean, they're becoming tubules. That's when we start referring to them as seminiferous tubules. Then the settled cells support and nourish the male germ cells. And that you need to understand that 300 million sperm cells are produced daily. Okay? Are produced daily. Okay, so this is spermatogenesis. Remember, we said spermatogenesis is also a type of cell division that is governed by the meiosis. So remember, we have meiosis 1, meiosis 2. So the cells usually start from a diploid cell. Then they go through the first primary spermatocyte. They go through the first phase of meiosis, which is prophase 1 of meiosis 1. So that's why where we make the primary spermatocyte. Then they undergo cell division, further cell division, and they become two type of cells. Okay, so this is the secondary spermatocyte that we have here, and this is still within meiosis 1. Then in meiosis 2, we find that the cells are now have 4 non-identical daughter cells, and that is where we have the developing sperm cells, which are haploid number and they have single chromatids. Okay, then the last part will be the sperm cell now, which will be formed, and it will also be an haploid number as well. So that is the spermatogenesis. Okay, so now in the process of spermatogenesis, there is a very specific and very interesting part which we call spermiogenesis. Okay, so in the process of spermatogenesis, there is an interesting part which we call the spermiogenesis. So spermiogenesis is by definition the final stage of spermatogenesis. The final stage of spermatogenesis, where around spermatids differentiate into mature spermatozoa. Okay, that's what you need to understand. So let me repeat. We are saying spermiogenesis. This word spermiogenesis. There's no. This is the correct spelling. Spermiogenesis. So spermiogenesis, we are saying it is the final stage of spermatogenesis, where the round spermatids differentiate into mature spermatozoa or the sperm cells. Okay, so and this process actually involves significant change in cell shape, structure, and as well as function. That's what we need to understand. Okay, so that's by definition. So now there are what we call stages of spermiogenesis. They are what we call stages of spermiogenesis, and for our literature here, we are going to talk about 6. So we're just going to talk about 6. So meaning literatures differ. There are some literatures we can say we have 4, 5, but here we are going to talk about 6. So the first stage is around spermatid stage. This is where the spermatid is spherical in shape. Then the second, so this is the first stage. The spermatid, this is the spermatid, and it is actually spherical in shape. You have the Golgi apparatus, the centriole, the Golgi region, mitochondrion. Then let's go to the second stage, which is the elongation stage. So the spermatids begins to elongate. So that is stage 2, they begin to elongate. Okay. Then stage 3, condensation stage. So in the condensation stage, the nucleus actually condenses. Okay, so in the nucleus begins to condense. So this the nucleus that is condensing. Okay, here it was like this, and here it is now condensing. Then stage 4, we have the acrosome formation stage, where the acrosome, which is an enzyme containing vesicle, now becomes formed. So that is the stage that you need to understand. So from here, the residue cytoplasm has actually gone, and you have what remained in front is the acrosome, which is on the tip. Okay. Then stage 5, we have the tail formation stage, where the flagellum or the tail actually develops. This is the, remember, after we formed the acrosome, we are now on the tail that has been formed. Then lastly, of the maturation stage, where the sperm becomes fully formed. This one. Okay, so these are some of the things you need to understand in the stages of spermiogenesis. Okay, so by definition and the stages, we are done with the spermiogenesis. Now let's look at the female reproductive system. So then this is the ovary. So this is the structure of the ovary. Actually, that's how it looks like. but we are more in actually learning about oogenesis in the reproductive system. Oogenesis. So oogenesis begins before birth actually. So on the primary, oocytes are present at birth. So females have what we call primary oocytes, so meaning they are actually born with a specific number of oocytes. So other literatures will tell you that they are born with 300,000 oocytes. Other literatures will also tell us to say they are born with 400,000 oocytes. But of course, since the literatures differ, we are going to say 300,000 to 400,000 primary oocytes. That's what are present at birth. Then the primary oocytes, they remain arrested in prophase 1 of meiosis 1. Okay, until of course puberty, when someone now becomes able to they go and continue there from prophase to another phase. So meaning before puberty, the oocytes are arrested in prophase 1. Okay. Okay, then let's go to puberty now. Oogenesis at puberty. So every month, 15 to 20 follicles begin to grow, passing through the three stages of the primary, the secondary, and the pre-ovulatory. The primary is also known as the pre-antro, the secondary is known as the antro, then the pre-ovulatory is the Graphian. Then you also need to understand that the development of an ovarian follicle is characterized by number one, growth and differentiation of primary oocyte. The other one is proliferation of follicular cells. The fourth one is formation of the zona pellucida. Then fourth, we have the development of the thicker folliculi. Okay, so this is what you also need to understand about the development of an ovarian. Okay, so we are going to start with the antro and the Graphian follicles, so in the primary and secondary phase. So in the primary oocyte, so after puberty, of course, it actually starts to complete meiosis 1 to form the secondary oocyte and the first polar body. So meaning in the antro phase during puberty, it just completes meiosis 1. Then it goes to form the secondary oocytes and the first polar body. Then the secondary oocyte is also becomes arrested in metaphase 2 of meiosis 2, and we will not proceed unless fertilization takes place. So meaning from the primary in the antrophase, we go to the secondary, which is the Graphian follicles. So which is the Graphian stage. Now the secondary oocyte in the Graphian stage, find that it will also be arrested in metaphase 2 of meiosis 2, and we won't proceed to another stage until fertilization takes place. So meaning if it does not take place, then of course it won't proceed, it will just be absorbed later on. Okay, that's what we are saying. Okay, so meaning the one which triggers the completion of meiosis 2 is actually the sperm cells. Okay, that's what you need to understand. Okay, so let's continue and see how the secondary or the follicle photomicrograph looks like. So this is how it looks like. The nucleus here, which is the sperm, the egg cell. Then they have the zona pellucida. Then you have the follicular cells outside it. And this is how we can draw it: the zona pellucida, the primary oocyte, and the follicular antrum. Yeah. Okay, so this is the secondary follicle. Then let's go to the ovary and the follicle. So this is the ovary which we are seeing here, and these are follicles. But of course, we are going to explain this. Okay, so at puberty, the females begins to undergo a monthly cycle of three stages. You have the proliferative, which is the follicular phase. Then you have the progestational, or also known as the luteal phase or the secretory phase. Then you have the menstrual phase. So meaning the first one which we start with is the menstrual phase, which lasts for an average of 5 days. Then you go to the proliferative phase, okay, which lasts for a period of 14 days actually, and it overlaps with the menstrual phase. Then on the 14th, there is ovulation E. Then after ovulation of the luteal phase, which lasts from 14 to 28, and it overlaps with the ischemic phase. Then after 28, of course, the cycle again begins again. Okay, that's the menstrual cycle. Then during the menstrual phase, superficial layers of the uterine endometrium are actually shed. So meaning the functional layer of the endometrium is actually shed. Then the basal layer is one which is going to remain. So endometrium has two layers. You have the superficial layer, which is known as the functional layer, which shades off during menses, and you have the basal layer which remains. Okay, that's what we are saying on this slide. Okay, so during the menstrual phase, we are saying the basal layer remains, whilst the functional layer shades off. Okay, then we have ovulation which occurs. So near day 14, we have what we call a stigma. Okay, the other name for the stigma in this case in embryology is a macula pellucida. Okay, that's the other name. So it's the one which appears on the surface of the ovary. Okay, so that's what's going to appear. So this same stigma, or known as the macula pellucida, in mammalian reproductive anatomy refers to the area of the ovarian surface. Okay, yeah, okay, on the ovarian surface. We have seen where the one which has started where it wants to rupture into somewhere. That's what we call a stigma, or on the ovarian surface, where the Graafian follicle will burst through during the ovulation and release the ovum. Okay, so the increased contractions of the ovarian wall extrude the oocyte with some follicular cells called the coronal radiata. What we're just saying is that, remember, after the stigma starts to have the follicular fluid, after it ruptures, the follicular fluid goes and attaches to the infundibulum of the tube. Then after that, then of course the cell will start coming out of the ovary into the infundibulum. Now, when it's coming out, it's actually, yeah, there's increased contractions of the ovarian wall. So it's actually excluded together with some follicular cells. Then those follicular cells we now refer to them as the corona radiata. Okay, so this is how they need to completely be transported to the infundibulum. Then there's the one which remains, the follicle one, the one which has remained will now be called the corpus. That's what we are saying. Okay, so remember we have remained with the corpus luteum. Okay, so this is what we were saying. So a ruptured follicle releases the egg cell, which is surrounded by cells, which we call the corona radiata. Okay, then we remain with the corpus luteum. So the corpus luteum is formed after ovulation by fascicularization of remaining granulosa cells and the thicker antenna. Okay, then this is a yellowish temporary gland that secretes progesterone. So the corpus luteum begins to produce a progesterone. Then what's the fate of the corpus luteum? So if no fertilization occurs, the corpus luteum begins shrinking about 9 days after ovulation and forms a fibrotic scar tissue called the corpus albicans. Okay, so that's what we have to understand. So this is if no fertilization but if fertilization does occur, then degeneration of the corpus luteum is prevented by the human chorionic gonadotrophin, which is an hormone secreted by the embryo, the HCG. Okay, so only prevents the degeneration of the corpus luteum. So meaning since its degeneration has been stopped, so meaning it continues to secrete progesterone until the end of the 4th month, when the placental progesterone becomes adequate and the corpus luteum degresses slowly. So meaning as long as the body does not have enough progesterone, the corpus luteum continues to produce progesterone until the end of the 4th week, when the placental progesterone becomes adequate. So it now becomes enough. Okay. Then that's only when the corpus luteum regresses slowly. Okay, so that the placenta can now start producing, can now continue producing its own progesterone. Now, before fertilization do occur, you have to understand that the spermatozoa reaching the oocyte must undergo number one, capacitation. Capacitation is the removal of the glycoprotein, okay, coat, the glycoprotein coat and seminoplasma proteins from the plasma membrane of the acrosome region of the sperm, so that they can cross the corona radiator. So meaning this is a sperm cell which contains chromosomes and the acrosome containing enzyme at the tip. So it has to go to the corona radiator, and the plasma membrane of the sperm is still here at this site. So what's going to happen is that there is going to be a removal of the glycoprotein coat, and the plasma proteins are also going to be removed, okay, during this stage. So it wants to cross the corona radiator. So once it crosses the corona radiator, we are in stage 3 now. Then that's where the acrosome reaction is going to take place. Okay, so the it's called the acrosome reaction. So the acrosome reaction, as the sperm binds to the zona pellucida, proteolytic enzymes are released to penetrate the zona pellucida. So in short, as we are saying is on stage 2, you're going to find that as the sperm binds the zona, in stage 3, as the sperm binds the zona pellucida, okay, proteolytic enzymes are actually released to penetrate the zona pellucida. That's what we are saying here, okay. Okay, so now that was before fertilization. Let's now look at the phases of fertilization. So phase 1, we have penetration of the corona radiator, which we have talked about before fertilization. Then phase 2, we have penetration of the zona pellucida, which we have also talked about. Okay, those are the phases of fertilization: the penetration of the corona radiator and penetration of the zona pellucida. Then phase 3 is now where we have the oocyte, stage 3 or phase 3. This is where we have fusion of the oocyte and the sperm cell membranes. So remember, after the chromosomes reaction, only the head and the tail of the sperm without the plasma membrane is going to enter the oocyte. Then the cortical granules in the oocyte release the lysosomal enzymes that bring about cortical ozonal reaction, which can cause number one, the oocyte membrane to become impenetrable to other sperm. So the oocyte membrane is one which, of course, as just below the zona pellucida, we have the oocyte membrane. So after one sperm goes in, it becomes impenetrable to other sperms. So in order to prevent polyspermia, then we have the zona pellucida itself. Also, the cortical reaction also causes the zona pellucida to alter its structure and inactivate the receptors for sperms, preventing further sperm binding. This is also what happens. Okay, so this occurs in phase 3. So this is the cortical or zonal reaction we are talking about. After one sperm goes in, then the oocyte membrane becomes impenetrable. The zona pellucida alters its structure. Okay, that's what you need to understand. So let me just do a recap. Okay, so number one started with the contact of the contacts. The second, we talked about the chromosomal reaction. Then the third talked about the growth of the chromosomal process. Then four, there's fusion of the plasma membranes. Then five, there's entry of the sperm nucleus. So mean the plasma membrane, of course, doesn't go together with the sperm nucleus. Then six, you have the cortical reaction now. Okay, so that's what you need to understand to form smaller cells, which we call the blastomeres. So they are loosely arranged until the 8th cell stage, where they undergo compaction. Okay, so meaning the two cells stage, they are going to be duplicating, but they are loosely arranged until they reach the 8th stage, where they undergo compactions, where they don't have to become loose. Then at day 3, they now reach 16 cell stage. So the 16 cell is called the molar stage, or the molar, which forms. The 16 cell molar actually forms composed of inner cell's mass, which is the future embryo proper, and the outer cell mass, which is the future trophoblast, which of course will form the placenta. Then all these form while traveling in the uterine tube towards the uterus. That's also what you need to understand. Okay, so we are saying we have the 2 cell stage. Okay, they became the 4 cell stage, 8 cell stage. Then at the 8 cell stage, you said is that they are going to undergo compaction because they were loosely arranged. Then they begin from 8, they go to 16. At the 16 cell stage, they are called the molar form, or the molar stage. Okay, and this is where the mass is composed of the inner cell mass, which makes the future embryo proper. Then you have the outer cell mass, which makes the future trophoblast, which of course leads to the placenta. Okay, so that's what you need to understand. Then, of course, there is no learning without visual aid. So this is a visual aid. Of course, the ownership is not for me. It's belongs to the Nucleus Medical Media. But of course, this is a very good video for understanding embryology. At least you are going to learn a lot and through the visuals. Okay, so let's start.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life. It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluids surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
Okay, so that was very good animation by the Nucleus Medical Media, and it's a very good one. It's helped me a lot to understand the first week, and hope you have also enjoyed it too. So of course, let's continue. So of course, we ended on the morula stage, where the 16 cell layer. So when the morula reaches the uterus, the fluid penetrates through the zona pellucida into the inner cell mass. So this forms a single cavity which we call the blastocoel, and the morula becomes a blastocyst. Then the inner cell mass, now called the embryoblast, and the outer cell mass called the trophoblasts. Okay, then the zona pellucida disappears at the 5th day, and the trophoblast cells at the embryoblast pore begins implantation in the uterine wall at the 6th day of the development. So remember, last time we ended on the inner cell mass will form the embryo part, and the outer cell part will form the trophoblast. So that's what we are just saying here. The inner cell mass will be the embryoblast, which is here. Then the outer cell mass will be the trophoblast. Okay, now we are saying the zona pellucida, the one which was on the outside layer, which is here, on the outside layer. is going to disappear on the 5th day. And after it disappears, then the trophoblastic cells are here at the embryoblast pore. They are going to begin their implantation in the uterine wall on the 6th day of development. So that's what we are saying. Okay, so let's now go to implantation of the blastocyst. The embryoblast cells gather at one pore, while trophoblast cells also form the wall of the trophoblasts. Okay, so you have the trophoblastic cells on the walls, on the lateral aspect, on the outer mass, forming the invading the wall. Then you have the embryoblasts also gathering at one pore as well. So this is what we are saying here. You have the embryoblast, which is here. Then you have the trophoblast cells here, and this is the uterine wall. And the trophoblastic cells are invading the uterine wall. That's what we are calling as implantation. Then the trophoblastic cells at the embryoblasts begin implantation in the uterine wall at the 6th day of the development, normally along the anterior or posterior wall. So this is what we are saying. Okay, of the embryoblast here, then the embryoblast on the other side, we have the trophoblastic cells. Trophoblastic cells are invading the uterine wall, as we have seen, and this is occurring at the 6th day of development. And it can either occur on the anterior or the posterior aspect. But this is the implantation of the blastocyst. And of course, again, like I said, embryology is a topic that is better understood with the animation. So of course, this is also another animation, very good one at that.
Cleavage and implantation. Following fertilization, the zygote undergoes repeated mitotic divisions called cleavage. This occurs while the zygote moves through the isthmus part of the fallopian tube towards the uterus. First cleavage is completed after 30 hours of fertilization. Zygote is divided completely into two. The daughter cells formed during cleavage are called blastomeres. Second cleavage is completed after 60 hours of fertilization. On the third day of fertilization, embryo reaches a count of 16 cells. The structure looks like a mulberry and is called morula. It is still surrounded by the zona pellucida and reaches the uterus around 4 to 6 days after fertilization. As the morula enters the uterus, it gets rich supply of nutrients. The outer peripheral cells enlarge and flatten further. They form a trophoblast or trophectoderm. The trophoblast cells secrete a fluid into the interior, creating a cavity called blastocele. With the formation of blastocele, morula is converted into blastula. This is called blastocyst because of the peculiar cyst-like arrangement of blastomeres. The blastocyst has a single-layered wall, the trophoblast, and a group of cells attached inner to it called inner cell mass. The zona pellucida disintegrates, facilitating the rapid growth of the embryo. The trophoblast then gets attached to the endometrium and the inner cell mass is differentiated into an embryo. After attachment, the uterine cells divide rapidly and cover the blastocyst. The blastocysts become embedded in the endometrium of the uterus. This is called implantation, which leads to pregnancy. Normally, implantation takes in about 10 days after fertilization.
Okay, so this is what I was talking about. It's very good that you need to understand. So this is a summary of the first week. So in short, we are done. So remember, I started from gametogenesis, but specifically when we started talking about the menstrual cycle, ovulation, the primary oocyte, secondary oocyte. Then we went to ovulation, the ruptured and released oocyte. The released oocyte entered the oocyte, entered the infundibulum. Then it was caused oocyte was penetrated by the sperm cell. Then the oocyte, of course, has to complete meiosis 2. Then after it completes meiosis 2, becomes a two-cell zygote, two-cell stage cell. Then on the 16th cell, then it becomes called the morula. From the morula, it is continue to divide and to actually have a cyst or have a cyst field. Then you call the blastola or blastocyst. Then it's still at this stage, it still has the corona pellucida, the zona pellucida. Then the zona pellucida disintegrates, which is going to facilitate implantation. So this is the summary of the first week of development. That's what you need to understand. Then we are okay. So what is the literature to use when you're learning embryology? So number one literature which you use is Langman's Medical Embryology, any edition, but of course 10 and above. So this is actually very good literature. Then of course the other one is also a good literature. So then you also have to know about the organization of Langman's embryology book. So there's the general and systems based embryology, but as most of the time we learn the general. I teach the generals to be specific. The system based is not so much. So just the general embryology alone. Then you have the typical chapter system, which consists of the main text, the clinical correlates, and the molecular regression. But me, I'm saying we are going to learn the general embryology. The systems, no. So in short, we are going to learn no development of the digestive system, no. That's not what how you'll be learning. You just learn the gestation periods alone. Okay. Then of course, through him all things were made. Without him nothing was made that has been made. And of course, thank you for watching. If you haven't subscribed, please subscribe and hit the bell notification. And see you on the next video. So this is our lesson 1 embryology. We have 5 lessons, so meaning the remaining 4 more lessons on embryology. So check out the channel playlist on human anatomy and physiology, and that's where you're going to find more videos on the embryology, and of course as well as other videos. Okay, thank you for watching once again. Please subscribe and hit the notification, and see you on the next tutorial.